here on this platform, we are giving a brief idea and brief knowledge about the topic of hemoglobinuria in sheep and goat, the causes and diagnosis. So we can say all that is red urine is not hemoglobinuria. On the basis of pathology, on the basis of the color and other things, we can discriminate between hemoglobinuria, hematuria, and myoglobinuria. Hemoglobinuria can be defined as presence of free hemoglobin in the urine. It is as simple as that. So it is associated with red color or amber colored urine, or the shades may vary from reddish yellow to dark yellow, and the urine is transparent and it remains pigmented even after centrifugation. So this is one of the criteria where we discriminate between hematuria and hemoglobinuria. In hemoglobinuria, the color remains the same as it was early, even after centrifugation. And in case of hematuria, once the centrifugation is done, the RBCs, they settle down, and the supernatant urine, it becomes colorless or light yellow to dark yellow, and at the bottom you get a button of RBCs. So this way you can discriminate between hemoglobinuria and hematuria. So it is a condition where the oxygen transport protein, which is hemoglobin, it is found in abnormally high concentration in the urine, and this condition, it is caused by excess intravascular hemolysis. And here the RBCs, they are destroyed and they release the free hemoglobin into the plasma. So since hemoglobin, it is filtered from the kidney, it is excreted into the urine, and this imparts urine a red color. Hemoglobinuria, it can also lead to acute tubular necrosis, which is the cause of uremia, and it is a cause of death, because hemoglobin molecule, it is a fairly big molecule. It cannot be filtered. Even it is filtered to a small extent, it is actually digested or it is absorbed in the proximal convoluted tubule, and only a minute amount is passed into the urine, which is negligible or which is unnoticed. But when this hemoglobin is excess in the plasma, all of it cannot be absorbed and it leaks into the urine and it imparts the red color to the urine. Hemoglobinuria, it is a pathological condition where the hemoglobin is passed into the urine. So hemoglobinuria, it usually occurs because of certain infectious or non-infectious disease, and it may also be because of deficiency or excess of certain minerals, and it may also be because of some metabolic disorders in the animal's body. So this will cause lysis of the RBC and release of the hemoglobin into the plasma, and this excess hemoglobin in the plasma, it is filtered through the urine and it causes hemoglobinuria. Since there is lysis of the RBC, so there will be anemia, because hemoglobin is the main molecule of oxygen transport to the tissue. So there will be anemia because of the rupture of the RBCs. There will be anemia, and because of reduced amount of hemoglobin in the blood, it will be hypoxia. It will lead to tissue hypoxia. So coming down to the etiological agents of hemoglobinuria. So first, it can be divided into two: infectious or non-infectious causes. Infectious causes like bacillary hemoglobinuria, eperithrozone, then babesia, leptospira, even Clostridium perfringens, it also causes hemoglobinuria. Then non-infectious causes, they may be either deficiency or excess. So deficiency, most important deficiency is of phosphorus, where you can see the hemoglobinuria. That is called post-parturient hemoglobinuria. And excess of certain minerals like copper, gossypol, then nitrates, nitrites, even the hypotonic solutions, hypotonic IVs fluids, that can also cause the hemoglobinuria. Even water intoxication can cause hemoglobinuria, or onion or ryegrass toxicity. All these cause hemoglobinuria. Even transfusion reaction, when there is anemia, we go for blood transfusion, and if the blood is not compatible, then there will be lysis of the RBCs, and this excess lysis of the RBC, again it will release the hemoglobin into the blood and it will lead to hemoglobinuria. pathophysiology of hemoglobin urea. Normally, hemoglobin, it is liberated from the circulating RBCs and it is converted. Some amount of the RBCs, they are lysed daily into the blood. RBCs, they do not have nucleus in them. This is because the RBCs are to hold a good amount of hemoglobin in them. This hemoglobin actually it binds the oxygen and the carbon dioxide in the blood. So while it is coming from the heart to the circulation to the tissues, it is bringing oxygen, and when it is going back from the tissues to the lungs and to the heart, so it is carrying carbon dioxide. So this uh, transport system is done by hemoglobin. So normally the RBCs, they do not have nucleus. They have a great amount of hemoglobin in them, so that the body can receive good amount of oxygen, and these cells can carry good amount of oxygen from them, through them. So this RBCs, since they are not nucleated, so they have a shorter lifespan. So they die or they are lysed within 120 days of their formation. So every day there is some amount of wear and tear, and these RBCs, they are lysed. When these RBCs are lysed, they will release the hemoglobin component. This hemoglobin cannot be floated free in the circulation, so it has to be bound with certain proteins, that is haptoglobin in the circulation, because this free hemoglobin is toxic to the body. So they are to be bound with certain proteins, that is haptoglobins in the circulation, and they are carried to the reticuloendothelial system. So this bound heptaglobin, it is cleared by the hepatocytes by formation of bilirubin, iron, and amino acids. And inside they are bound, they are taken up by the macrophages, that is Kupffer cell in the liver. So if there is excess amount of hemoglobin, because the body circulation has a limited amount of haptoglobin, so whenever there is hemolysis, the hemoglobin is bound to the haptoglobulin, and when this haptoglobin attachment or the hemoglobin combination with the haptoglobin is saturated, this haptoglobin, it is saturated, and this hemoglobin, which is now free, it is accumulated in the plasma, and this is called hemoglobinemia, because there is excess amount of hemoglobin in the circulation. And now this hemoglobinemia, or this plasma which is having huge amount of hemoglobin, that passes through the glomerulus, and it is filtered. But when it is filtered, some amount of this hemoglobin, it is absorbed by the proximal convoluted tube cells, and it is again degraded. But if the capacity of this system is also exceeded, then this hemoglobin, it is released into the urine, because it crosses the renal threshold, and it passes into the urine, leading to hemoglobinuria. Now this hemoglobin, it precipitates in the form of cast in the tubule, because this excess hemoglobin which was filtered, actually when this hemoglobin it attaches with the haptoglobin, the molecule becomes big in size, and it cannot be filtered through the glomerular apparatus. But in case of excess amount of hemoglobin in the urine, some amount is plugged into the renal tubular cells, and this causes uremia. And this uremia, it is one of the chief cause of tubular nephrosis and death, and uremia in case of patients. Now we'll study one by one causes in detail. So, bacillary hemoglobinuria, Clostridium perfringens, then erythrocytosis, then babesiosis, and leptospirosis. These are the important infectious causes. Other infectious causes are also there, but these five important diseases which cause hemoglobinuria in case of sheep and goat. Bacillary hemoglobinuria. It can be defined as it is an acute fatal disease of sheep and cattle. So goat are also affected, but sheep is more afflicted with this. And it is characterized by hemoglobinemia and hemoglobinuria. As it is indicated by this name, there is bacillary hemoglobinuria. So hemoglobinuria has to be there. Okay. So this is a disease of sheep and goat and cattle. There are many factors which cause hemoglobinuria. One important diagnostic factor in this case of hemoglobinuria, bacillary hemoglobinuria, is the season. Actually, it is more common in summer and autumn, and this is particularly seen in at the time when the fluke population is more in the animals. So whenever there is endoparasitic infection, and whenever there is excess of starvation, when the uh, animals immune system goes down, so that time you will find this hemoglobinuria, and you can to 80% you can diagnose it as because of Clostridium hemolyticum, because of the season and because of the prevalence of flukes in that particular time period. So what happens, the flukes, they are the eggs of the flukes, they are present, the animal, they go, they take this snails, and this metasaccharide enters into the body and it migrates to the liver. Now this fluke, it develops in the liver, in the bile canaliculi, and in the liver hepatocyte, in the hepatic tissue. So this, while traveling, it creates a path which causes damage to the cell or to the tissue. So this Clostridium hemolyticum, it is there in the body, and along with the fluke, along with the track of the fluke, it goes and it settles, and since it is getting a nutritive media of the damaged cells, so it will proliferate on that, and it will lead to damage to the liver and to the Kupffer cells, and this migration of immature liver flukes and other parasites also, that causes the actually that acts as a nidus of um, proliferation for this Clostridium hemolyticum bacteria, and there will be necrotic hepatitis, and there will be parenchymal damage, and the bacterial spores, they start germinating, and that lead to the hemoglobin urea. So this bacteria, as I said, it proliferates and it releases the exotoxins. Clostridium have that tendency to release toxins, so they release exotoxins, including the beta toxins, phospholipase C, and this causes hepatocellular necrosis, and this also leads to intravascular hemolysis because of shortage of certain types of proteins, amino acids in the blood, which are synthesized by the liver. Since the hepatic tissue is damaged, these amino acids or these proteins are not being formed there, and that leads to hemolysis, and this again hemolysis leads to anemia and hemoglobinuria, and this is the characteristic of this disease. So migratory tract of the fluke actually precipitates the disease. So while diagnosis, we can go for identification of the fecal sample. If you are getting hemoglobinuria, along with that, if you are getting fluke eggs, so 80 to 90% diagnosis is that this is because of, and if there is some pain in the abdomen, especially on the right side of the abdomen where the liver is situated, so you can palpate the animal, and because the animal will elicit pain, and if you can see a enlarged liver which can be easily palpated at the hypochondric region and ahead of the last rib. So the liver is enlarged, actually liver's anatomical position is hypochondric area, but in case of enlarged liver, you can see a much amount or much mass of the liver, it is beyond that margin. So one thing is pain in the abdomen, then you can find the eggs of the fluke in the fecal sample, and then hemoglobin urea and the season. Along with that, you can culture the organism. If you can take a biopsy of the liver, you can do that, and even if you can go for culture of the fecal sample, you can get the Clostridium organism. So this is about the diagnosis of bacillary hemoglobinuria. This is a liver which was afflicted with the liver fluke, and it had bacillary hemoglobinuria. So you can see multiple tracts of the liver and necrosis of the liver everywhere, and thickening of the bile canaliculi. See here, the periphery of the bile, bile canaliculi, it is whitish in color. That means it is fibrous. Usually it is hollow structure. But when you see a peripheral ring of white color, that means there is calcification and fibrosis. So this is a typical liver of bacillary globinuria affected liver. So another organism in the same group, that is Clostridium, it is Clostridium perfringens. So here also you can see hemolysis, intravascular hemolysis. As there is hemolysis, the signs which are associated with reduced amount of hemoglobin in the blood, they are icterus, then there is anemia, tachypnea, and there will be the animal will be very weak, depressed, and there will be recumbency. So here, for the positive diagnosis, you can go for identification of the or isolation of the toxin with the help of ELISA test. So alpha toxins you can easily make out with this toxin and this ELISA test. And you can also go for molecular techniques. Molecular techniques are very common these days, and they are very precise and very sensitive. So since we are talking of diagnosis at farmers doorstep, where we don't even have microscopes many times, so basically you can differentiate on the basis of, as I said, bacillary hemoglobinuria, there will be enlarged liver, and you'll find flukes. Many a times the animals are grazing around some marshy area or near ponds or lakes. There you find snails, and from these snails, the through the pasture, the animal they get the fascicular infection, and because of that there will be bacillary hemolytic infection. Here you have to see whether the season is there and all, and if the animal is vaccinated or not. If you bring the blood sample in the laboratory, you will typically see neutrophilic lymphocytosis, because immature leukocytes they will be coming, especially the neutrophils will be present in the blood. So that is a typical finding of Clostridium perfringens. Treatment of Clostridium, either it is Clostridium hemolyticum or Clostridium perfringens, you can go for administration of long acting penicillin. When there is acute case, you can go for treatment with Clostridium antitoxins. Etiology of eperithrozoonosis. Now it is classified under bacteria Mycoplasma. So Mycoplasma ovis, it is the causative agent. Here there is again fever, because since it is a Mycoplasma organism, and it will definitely cause some amount of respiratory problem. It will also lead to hypoxia, and there will be anemia, and there will be hemoglobinuria because of hemolysis. Okay, so the diagnosis here, here you can go for blood smear examination. In blood smear, typical finding of Mycoplasma is that there will be regenerative anemia, and finding of intra-erythrocytic bodies. And if the animal is chronically infected, then there will be high amount of nucleated RBCs, that is reticulocytes. That is very peculiar picture of eperithrozoonosis on the blood smear. Since this organism, it consumes good amount of glucose for its proliferation and growth, so the animal, there will be hypoglycemic. The animal will be hypoglycemic, and because of that there will be metabolic acidosis also. So while checking for the organism, and if you find a symptom that the animal, they are showing symptoms of lethargy, weakness, hemoglobinuria, along with that, because of hypoglycemia, there will be conversions. So this can also be seen in case of animals. A few animals will show convulsions, and there will be death. But this is a chronic disease, so you should differentiate between eperithrozoonosis or this Mycoplasma, and many times you find these symptoms in case of trypanosoma infections also. Sudden there will be convulsion, there will be frothy salivation, and the animal falls and dies. So this peculiar symptom is seen in case of trypanosomiasis also. So definitely for confirmative diagnosis, you have to collect the blood sample, prepare blood smear, and go for staining and observing the parasite in the blood smear. You will find the organism in the erythrocyte, that is intra-erythrocytic parasite you can find, and you can also go for post-mortem lesions because this organism, it consumes a lot of glucose and there is lysis of the RBCs. So the tissues, mucous membrane, there will be pale in color. Along with that, since there is lysis of RBC, there will be icterus and splenomegaly. Why? Because when there is hemolysis, the RBCs, dead RBCs and the material, it goes into the spleen for processing. So this can be seen, and along with that there will be vasculitis, and since there is reduced RBC, so there will be reduced osmolality of the vascular circulation, and that will lead to tissue exudation and edema and in different cavities. So this is a typical smear of the eperythrozoon parasite, wherein you can see 2 to 3 dots, reddish dots in each RBCs. So this is a typical feature and slide of eperythrozoon infection. And here on the right side, you can see a pallor mucous membrane of the conjunctiva. Babesiosis is very common. The infection is intra-erythrocytic protozoan parasite. That is Babesia ovis in case of sheep. So sheep and goat. So in case of babesiosis, the history will be tick infestation. So tick infestation is very common, and because of that, there will be hemoglobinuria. And when you prepare slide, you will find the intra-erythrocytic protozoan parasite in the slide. So clinical signs, definitely there will be coffee colored urine, and because there is reduced osmolality, there will be tissue exudation, and similarly there will be edema in the lungs, that is pulmonary edema, and this will lead to mucopurulent nasal discharge and coughing and dryness, dyspnea, and icteric sclera because of hemoglobinemia, increased amount of bilirubin in the body. There will be icteric sclera and mucous membrane. And since the animal is weak because of reduced RBC, all the tissues are compromised, that they are not getting oxygen, so they will not get enough amount of energy. So the animal will be lethargic, there will be reduced amount of energy, and because of this there will be reduced abdominal motility also. This will cause bloating of the abdomen, and because there is bloating and there is intestinal stasis, you can call, not complete stasis, but there will be reduced movement. So when there is reduced movement of the bowel, all the ingesta, it starts putrefying, and because of that there will be diarrhea. And since there is huge amount of hemolysis and there is huge amount of vascular damage, so there will also be oozing out of blood from the capillaries. So the capillaries in the intestinal tract, they will also be ruptured, and this will lead to blood in the GI tract, and this will lead to hematochezia. So diagnosis, again a history of tick infestation, then there will be increased temperature. In case of eperythrozoon or Mycoplasma ovis, the temperature will not be a prominent symptom in many cases because Mycoplasma infection, it is long standing, so you may get or you may not get pyrexia. But here definitely in protozoan parasitic infection, you will get pyrexia because of reduced amount of hemoglobin, reduced amount of oxygen. There will be feedback to the brain to get more amount of oxygen, so the heart will start beating more to draw more amount of oxygen from the pulmonary vein, and in turn there will be increased amount of respirations to get more amount of oxygen for oxygenation of the blood, and that blood, oxygenated blood, has to be reached to the heart, and from there it is to be redistributed to the tissue. So when tissues are getting less amount of oxygen, there will be definitely tachycardia, tachypnea, and there will be prescapular and prefemoral lymphadenopathy in case of babesiosis, because this organism, it causes inflammation and it leads to increased amount of defense mechanism cells like neutrophils, macrophages. They have to come to the circulation to protect the body. So there will be lymphadenopathy, and again there will be respiratory diseases because there is reduced oxygen, reduced osmolality. So tissue fluid, that is the amount of fluid from the capillaries or the vascular bed, it will leak to the extra capillary space, and this will cause edema in the lung, and this will lead to respiratory wheeze. Now, the laboratory diagnosis, again, anemia, neutrophilia, thrombocytopenia. This is one of the important finding in case of Babesia. In case of Ehrlichia also, you find thrombocytopenia. So, there will be hypoproteinemia, and because everywhere tissue damage is there, so is the liver damage, and that will lead to hypoproteinemia. And because RBCs are ruptured, there will be hyperbilirubinemia, and again RBCs are ruptured, so there has to be hemoglobinuria. This Babesia, it has a characteristic feature of settling into the kidney tissue cells. Okay, again the Babesia, it goes and infects the um, nephrons, and it settles there, and it elicits antigen antibody reaction there. So there will be again a problem in the nephrons, and this will cause certain amount of or huge amount of damage to the renal parenchyma. And because of this, there will be, because of the uremia, because of hyperbilirubinemia, because of hyper hemoglobinemia, and because of damage of the nephrons directly by the Babesia organism, because of vascular, because it is entering into the vascular compartment also, so it will damage there also, and vascular endothelium also. It will cause thrombosis or thromboembolism in the nephrons in the body, in the minor capillaries in the body, especially the nephrons. So the organism, it causes damage to the kidney, and it leads to renal failure. This is a very peculiar finding in case of Babesia. So whenever we try to eliminate the organism, we eliminate the organism from the circulation, but the organism, it goes and it settles in the spleen and in the kidney. The Ehrlichia, it settles in the spleen for long times, for many years, and the Babesia, it goes and settles in the renal parenchyma, renal tissue. So to eliminate infection from these tissues, it is very difficult. Because of this, whenever you go for identification or you go for diagnosis of these organisms, even if the organism is not present in the circulation, the organism only appears in the circulation once the immunity of the animal goes down, then this organism, it gains upper hand and it enters into the circulation. But otherwise, it is there sitting hidden in these deeper tissues. So when you go for applying PCR, definitely you will get the antibodies and some amount of antigen in the blood smear, blood, if you go for diagnosis with the help of PCR. So real time PCR, it is more important than the other methods. And along with that, you can go for blood smear staining. You can find pear shaped organisms, and you can also see the signet ring shaped organism in the blood smear in the erythrocytes. Now, leptospira is another causative agent for hemoglobinuria, especially in sheep and goat and in cattle also. It is a zoonotic disease, or this organism, it is found in the sewage, in the urine of the rodents and all. So you can find this organism in the rainy season. The leptospira are caused by different serovarieties of the organism leptospira. So hemoglobinuria, it is a prominent feature of leptospira infection. Now clinical symptoms, there will be intravascular hemolysis and there will be sepsis. So definitely there will be high rise of temperature, and because of temperature there will be depression, and since there is hemolysis, again there will be dyspnea, there will be there will be edema, and since there is huge amount of hemolysis. RBCs are reduced, oxygen carrying capacity in the body is going down, and there will be exercise intolerance, weakness, and anemia. Again, because of hyperbilirubinemia and hyperhemoglobinemia, there will be icterus and death in the animal. Important finding in case of leptospira in sheep is the sheep, it may roam around freely, and some sheep you will find dead suddenly. They are walking and they'll fall down and they are found dead. And then, since there is reduced amount of oxygen, vasculitis and all, so there will be abortion and stillbirth. The animal, the fetus will not grow, or the and fetus will be born dead. So the leptospira, it also goes and it gets entry into the tubules and into the vascular bed in the kidney, and it gets localized there, and it causes damage to the kidney, and you can see mottled kidney appearance. There will be necrotic patches on the kidney cortex and in the medulla also, and those necrotic patches, they can be seen from the capsule also. Even if you don't remove the capsule of the kidney, you see that mottled appearance of the kidney. This is very peculiar finding in case of leptospira. Now, diagnosis, you can go for, along with these symptoms, you can go for identification of the organism in the blood and in the urine, especially in the urine. For this, you need a dark field microscope. Cannot be seen directly from a microscope, ordinary microscope. You need a dark field microscope for that, and you can see the organisms. They are moving like a screw. They are moving like screw. They are elongated spirochetes and they move like screws. Typical finding in case of leptospira. Along with that, the urine is dark, and along with that, the bladder and kidney, they are also full of this dark colored urine. Culture of this organism is very difficult, and since it is zoonotic, so it is to be handled with care, and it needs special media. So we can send these samples to the Madras Veterinary College, and you can find the you can get the organism cultured there. Treatment, broad spectrum antibiotics. Now, how to prevent? So, in endemic area, you can go for vaccination. Now, move on to non-infectious causes. Most important and the only important cause of deficiency is phosphorus deficiency. It's called hypophosphatemia or post-pasturant hemoglobin urea. So post-parturient hemoglobin urea, it is very common in dairy goats also, fairly common in sheep. So this anemia appears because of the decreased ATP production in by the RBCs or inside the RBCs, and because the phosphorus is an important component of the phospholipid membrane of the cells. So the RBCs are being lysed because of very fragility of the RBCs. So post-parturient animals, they are more susceptible because definitely the animals are prone to hypocalcemia, hypophosphatemia because of drainage of mineral for the development of the fetus. And along with that, again, drainage of those minerals in milk during initial stage of milking in these animals, because after parturition there will be huge amount of milk secreted by the animal for the calf, so the minerals are being drained out in the milk, so definitely there will be deficiency of these minerals. Now copper deficiency is also there, which may contribute to this severity of this anemia in case of hypophosphatemia, and that also causes lysis of the RBCs, and because there are certain enzymes which need copper, and these enzymes are important for oxidation, for antioxidant properties in the body. So when there is reduced copper in the body, these antioxidants produced in the body are reduced, and so all the cells including the RBCs, they are susceptible to the oxidants, and there is lysis of the cell, there is death of the cell. Next, sir. So again, let us come to the causes of hypophosphatemia. Uh, Hypophosphatemia may be because of reduced gastrointestinal absorption. The animal may be taking good amount of food. The owner may be feeding good amount of food to the animal, but because of reduced absorption from the gastrointestinal tract, and especially the phosphorus is absorbed from the duodenum, the the small intestine. So any damage in the small intestine, or because of excessive acidic pH and malabsorption from the GI tract. Again, as we say, because of acidity, there will be gastric upset, and because of that, there will be ulcers, there will be problem in the gastrointestinal mucosa, and that will lead to malabsorption. And along with that, if the animal is being fed a diet which is high in phosphates, okay, so phosphate binders that will also not allow the phosphorus to get absorbed from the intestinal epithelium. Okay, so this is one of the reason of hypophosphatemia, that the food is very high in phosphate binders, or there is some problem in the gastrointestinal tract, or because of the reduced pH of the gastrointestinal tract, there will be reduced absorption of the phosphorus, and that leads to malabsorption of the phosphorus from the intestinal tract. The second reason is because of increased urinary excretion. Because again, in urinary excretion, it depends on the presence of vitamin D. If there is vitamin D deficiency, the phosphorus, it will be excreted, it will not be absorbed from the renal tubules. Okay, so there will, because of hyperphosphatemia of parathyroidism also, there will be more amount of phosphorus, and that will be removed from the kidneys and from the urine. And again, gastric disorders also, that will also lead to, actually, phosphorus is absorbed from the gas intestinal mucosa through a sodium phosphorus co-transport system. If there is any problem in the intestinal mucosa, this sodium phosphorus pump, it doesn't work properly, and this vitamin D, it is a loop actually, or it is a next stage of transporting the phosphorus and calcium to the bone, to the storehouse. So if this co-transport, sodium phosphorus co-transport, is not working properly, the phosphorus is not being absorbed, and when the phosphorus is not being absorbed, the vitamin D will not deposit the phosphorus in the bone. Again, if sodium phosphorus pump is working properly, but if the vitamin D deficiency is there, again this will not be transported, and there will be hypophosphatemia. So again, gastric disorders, then renal disorders, they all decrease the absorption of phosphorus. So phosphorus is absorbed from the proximal convoluted tubule. So this is not being absorbed, and this leads to phosphorus deficiency in the animal. Now, third reason is defective internal redistribution, because this is again syndrome which is called refeeding syndrome. Suppose someone is being starved, if the animal is being starved for a long time, and if it is offered a good amount of food, again there will be excess of certain minerals, there will be deficiency of certain minerals. Deficiency will be okay, but excess of certain minerals, that will not allow certain minerals to get absorbed. Okay, so this is called refeeding syndrome. Then hungry bone syndrome, since the phosphorus is not in proper level in the blood, so there will be more amount of breakage of bones for maintaining the normal amount of phosphorus in the blood. Okay, but again, there is osteoclastic activity to bring down the phosphorus in the blood, but the bones, they are not being restored with the amount of loss. So this is called hungry bone syndrome, and this is because of hypocalcemia. And along with that, during the treatment of ketosis with insulin therapy, with the insulin therapy, the phosphorus is being also transported in the intracellular space. Okay, so the phosphorus level in the blood it goes down. Okay, and the phosphorus, because insulin is taking glucose, the energy source, and it phosphorus is also a source for the ATP molecule. No? So the another phosphorus is being consumed by the ADP for formation of ATP and for formation of energy, through which this our body or the cells will work. So because of ketosis, and when there is ketosis there will be a reduced absorption of glucose. When you give insulin therapy, that will help in moving the blood from the moving the glucose from the blood to the intracellular space. So again, during treatment of ketosis and during insulin therapy, there will be again deficiency of phosphorus and respiratory alkalosis. All these things, they again lead to phosphorus deficiency. So phosphorus deficiency is because of GI absorption, or because of increased excretion, or because of any defective internal redistribution system, like bones. From bones the the phosphorus is being uh, transferred to the circulation for maintaining the normal phosphorus level. But again, because of hypocalcemia, again because of certain hormone deficiency, because of certain enzyme deficiency, the broken amount or the taken away amount is not being restored properly. Okay, so these are the main three uh, reasons for hypophosphatemia. Next, sir. So, pathophysiology of hypophosphatemia. As I said, this phosphorus is an important component of phospholipid membrane of the cell. So, because of decreased level of phosphorus, what will happen? There will be membrane fragility, and there will be increased fragility of the RBCs. And these RBCs, since they become more fragile, they easily get destructed, and there will be intravascular hemolysis. Next, again, the phosphorus is a component of ATP molecule. So, because there is a decreased amount of ATP molecules in the body, there will be decreased energy level in the body, and this results in fatigue, muscular weakness, and reduced muscular contractility of the cardiac muscles. So, there will be fatigue of all the cells in and all the muscles in the body. So, the animal will be dull, depressed, and lethargic. Now, then again, as I said, bone metabolism, phosphorus is an important component of bone. So, because of decreased level of phosphorus, there will be hypophosphatemic rickets and osteomalacia. Okay, many times when there is deficient, when we see rickets, we jump to a conclusion that the animal is having reduced calcium level. So we go for administration of vitamin D3, we go for administration of calcium without even knowing that the amount of calcium in the body is normal. But many times it also happens that because of some gastric upset, gastrointestinal problem, the calcium and the phosphorus, it is not being absorbed, and because of that the bones, they become fragile, and they easily break, or the costochondrial junction, it gets enlarged, and it leads to hypophosphatemic rickets. And this is particularly seen in old age, that is osteomalacia, and the bones, they become very very weak, and there is pain in the bone. Okay, now another thing is that this phosphorus, it is an important component of diphosphoglycic acid, and this 2,3 diphosphoglycic acid, it is an important enzyme. Actually, what it does, when the hemoglobin, it is there in the RBC, and when the oxygen in the blood, in the lungs, it is the this enzyme which actually combines the um, oxygen with the hemoglobin. Okay, so decreased production of this enzyme, what will it cause? It will cause reduced release of oxygen from the hemoglobin, because this hemoglobin is tightly bound to the oxygen with the help of this enzyme. If this enzyme is reduced, sorry, if this enzyme is reduced, then the affinity of the hemoglobin to the oxygen is increase, and this will not allow the oxygen to dissociate from the hemoglobin. Okay, so what will happen, even though the hemoglobin is full with oxygen, but because of the deficiency of this disease, it is not getting dissociated at the cellular level, at the tissue level. So the oxygen, though oxygen level and oxygen concentration in the blood is good enough, but there is tissue hypoxia. It is because of reduced amount of this enzyme. Okay, so the enzyme level has to be checked, so that the when the oxygen molecule or the hemoglobin and oxygen molecule, they are reaching the tissue, where the amount or the partial pressure of the carbon dioxide is more at the tissue level, at the venous exchange. So this oxygen molecule should dissociate, and the hemoglobin should combine with the carbon dioxide. But in the absence, or in the deficiency of this enzyme, this hemoglobin molecule, it cannot release the oxygen molecule, and thereby the carbon dioxide concentration at the tissue level, it remains the same, but oxygen is not being released there, and this will cause hunger, oxygen hunger of the tissue, and lead to tissue hypoxia. Later on, there will be tissue necrosis. Now, toxicosis and poisoning reasons. So, again, water intoxication, too much amount of water, it will lead to dilution of blood. So the RBCs, they will withdraw more amount of water, they will swell and they will burst, and this way there will be lysis of the RBCs. Again, the hypotonic IV fluid, they also work with the same mechanism, that hypotonic, because the inside the cell there is hypertonicity compared to the extracellular space. When you are giving more amount of hypotonic fluids, then this fluid is being absorbed by the cells, and especially the RBCs, they swell and they rupture. Then onion and ryegrass toxicity, it is very common in sheep. And then copper poisoning, it is one of the important causes of hemoglobinuria in sheep. Then acute lead poisoning, nitrate or chlorate toxicity, gossypol poisoning, and transfusion reaction, where the blood is not compatible. So there will be lysis of the RBCs, and this will lead to hemoglobinuria. Copper poisoning. So as I said, sheep are more susceptible than the goat. So breed wise, also it is the susceptibility, it varies. But as compared to acute toxicity, chronic toxicity is more common in sheep and goat. So some breeds like merino sheep and all, they are more prone to copper absorption and storage problem, and this leads to the accumulation of copper in the body, in the cells, and this leads to lysis of the cells. Other breeds of goats, they are more resistant to this, and they're more prone to deficiency. As I said, copper deficiency also causes hemoglobinuria, and copper poisoning or copper toxicity, it also causes hemoglobinuria. So acute poisoning, it follows if the animal takes 20 to 100 milligram of copper uh, per kg copper per day. Okay, and primary copper toxicity, it is seen in sheep when there is excessive amount of copper intake for a long period of time. Actually, this copper, when it is taken in huge quantity, all of a sudden there is acute toxicity. But if it is taken over a long period, then this copper is getting absorbed, it is getting stored in the liver, and later on it is released, leading to chronic copper poisoning in sheep. So if the goat, if the sheep and goat, they are taking 3.5 milligram per kg of copper daily while they are grazing on the field, usually copper sulfate is sprayed as a acaricide, especially in fasciole infested area. Copper sulfate spraying or top dressing is recommended. So if sheep are grazing on such pasture, if you have sprayed the copper sulfate and you have let loose the sheep on the same field, then before the snails they are killed, the copper is being taken by the sheep and it leads to copper poisoning. Okay, copper toxicity. And even if the copper is incorporated in the feed in the concentrate mixture at the rate 15 to 20 parts per million, that also causes copper toxication. And if a particular soil is deficient in molybdenum and sulfur, that will also indirectly cause copper accumulation and copper poisoning. In sheep, copper to molybdenum ratio, it greater than 101, it leads to accumulation of excess copper in the blood and in the body tissues. So decreased formation of copper molybdate or copper sulfide, this complex, it impairs the excretion of the copper in the urine and the feces. So the sign of toxicosis, they develop when water is even supplied from a copper plumbing parts. If the tap is of copper and it is rusted, and so that time also you can find copper toxicity. Now, important source of copper toxicity in case of sheep and goat is like trace minerals. And also sometimes what happens in a farm, in mixed farming practice, where we have cattle also, we have a sheep, goat also, we have horses. Few people they have this, that they'll have even fish, aquaculture, then they will have poultry. And if you are formulating a feed for a particular species and the same mineral mixture is being fed to sheep and goat or other animals, not for this particular, in case of copper we are talking of sheep and goat, but for other minerals also, the mineral intake is species specific. If you are giving more amount of particular mineral to a particular species which is not required for that species, then it will definitely cause toxicosis. And then that is why the feed formulation has to be taken care of very carefully. So this disease, as I said, it remains subclinical until a huge amount of copper it is accumulated in the liver and in the bloodstream. And now this increased liver amount of copper in the liver, this will cause increased enzymes, and this will cause later on this will lead to fragility of the RBCs and rupture of the RBCs and lead to hemoglobinuria. Now serum concentration of copper, usually if it is taken in huge amount in sheep and goat, all of a sudden then there will be acute rise, and this will cause lipid peroxidation and lead to intravascular hemolysis. This hemolytic crisis, again it will be precipitated by n number of factors. Like suppose there is a toxicosis of copper, but it is going unnoticed. But all of a sudden, if there is any change in the weather, if there is any transportation stress, or if there is handling problem, handling stress by the owner, and oh, sorry, to the sheep by the owner while sharing and all, or suppose there is some pregnancy, lactation, and all these factors, they will lead to. And even if the sheep they are being chased by a dog, that will cause sudden stress to the sheep are very delicate animals. So even if these animals are being chased by dogs, and if there is copper toxicosis which is going unnoticed or it is in subclinical stage, all of a sudden it will flare up, and the sheep it will fall down suddenly and there will be death. I was saying about one problem in sheep, that sheep are very delicate, and all of a sudden when the sheep they are being chased, they will fall down, they will become stiff, and they will die. So this is the reason for death, sudden death of sheep. So sheep, if they are injected with soluble form of copper, they also develop acute sign of uh, copper toxicity, and there will be hemolysis. And sheep, the affected sheep, they are depressed. Again, all the signs of anemia because of lysis of the RBCs, there will be signs related to anemia, and there will be depression and dullness, and there will be diarrhea in the copper toxicity cases in case of sheep. And again, as I said, many animals, they will be found dead with hemolysis and icterus. You will not know, suddenly the in the morning the animal was okay, and while grazing, suppose there was some stress, or suppose there was chasing by the dogs or some wild animal in the forest area, and these animals, they will fall down, there will be hemolysis, there will be hemoglobinuria also, and there will be death. And when you go for post-mortem examination of such animals, you will find erosions and ulcers in the abomasum. This is a very peculiar. So here again I want to make a point to remember that abomasal ulcers are also signs of thylariasis. So again, here you have to discriminate between whether it is a case of thylaria or whether it is a case of abomasitis because of copper poisoning. So this acute disease, there will be a sudden release of the copper from the liver in large amount, and that will lead to acute toxicity. So this is a post-mortem picture of the chronic copper poisoning case in case of sheep. You can appreciate that the in the left top picture, I have actually pasted one picture about the another. So there will be increased icteric membrane, there will be, okay. So there will be a yellow, dark yellow color discoloration of the conjunctival mucous membrane, that you can appreciate very well. And in the lower picture on the same slide, so there will be, see, you can see icteric mucous membrane, then serosa will be icteric. And in the right side of the same slide, you can see the kidneys and the liver. Kidneys, they are bronze color, that is gun metal type ka color rehta hai. So that will be bronze discoloration of the liver and the kidneys. And if you open the kidney, you will find a typical brownish, bluish brownish discoloration. So on the presumptive diagnosis, on the basis of sudden death and hemolytic crisis, you can suspect that this is a case of some poisoning. And what poisoning? Again, we can take the history, that whether this animal was in stress, or when you cut open the carcass of such animal, you will find extensive liver damage with the bronze discoloration on the liver. Then you can also go for collection of the blood, and you can see the copper concentration in the blood. Then usually in the blood, if you are giving a if you are taking blood sample randomly in live animals, the increase in the copper is very transient. Copper level, blood copper level increase is very transient, so many times it goes unnoticed. So from the sample of a dead animal, or even you can go for biopsy of the liver and kidney at the antemortem examination. You can take the biopsy samples, and you can go for estimation of tissue copper concentration of the liver and the kidney. That will give you a correct picture. Then evidence of blue green ingesta, that is a very important point. And increased fecal copper concentration, that is again an important finding in case of copper poisoning. So whenever we go for postmortem examination, whenever we suspect for some kind of poisoning, we definitely send the sample to toxicology lab. And while sending the sample, we can write a note that suspected for copper poisoning. By seeing the liver, by seeing the kidney, and by seeing the ingesta color, we can find out that this is a case of copper poisoning. So we can go and we can send the sample to toxicology laboratory, and we can get the correct diagnosis. So in case of chronic copper poisoning, the blood and the liver copper concentration, they are definitely increased. And during the hemolytic crisis, definitely they are increased. So, what are the clinical pathological changes? Deposition of the hemoglobin in the renal parenchyma, in the tubules. As I said, that hemoglobin deposits as casts in the renal tubule, that to proximal convoluted tubule. So there will be renal impairment. So definitely you will find the increased creatinine level, increased blood urea nitrogen level. Then liver enzymes, they will also be increased. Then serum copper concentration, it will go high as 5 to 20 milligram per ml. And then liver and kidney, again the copper concentration, it exceeds 500 milligram per gram dry weight of the tissue, or and 80 to 100 milligram per gram dry weight of the kidney tissue. Then you can give a confirmative diagnosis. Then tissue discoloration is also there. More amount of methemoglobin is seen rather than icterus here, and this is because of chronic copper poisoning. Again, there will be swollen blue, black, or gunmetal colored, bluish brown, that is gunmetal colored kidney, that can be seen. And when you cut open the kidney longitudinally, the renal cortex, it may be demarcated from the, very well demarcated from the medulla, because copper is deposited in the tubules. So the glomerular portion will be spared, and there will be clear discrimination between the colors of the medulla and the and the cortex. So there will be enlarged spleen and dark brown black color parenchyma, which is again indicating hemolysis. Because of hemolysis, the product byproduct of the RBCs and the and the hemoglobin, they are reaching the spleen, and because of that, the spleen is getting engorged, and that will lead to splenomegaly. And the liver, it will also be friable, it will be enlarged, and it will be orange or yellowish in color because of hemolysis. Again, there will be increased amount of bilirubin, and this leads to yellowish brown discoloration of the liver. Then if you check the urine, that will be port wine colored urine or dark brown colored urine, which is again suggestive of hemolysis. If you go for histopathological examination of the liver, and the kidney, then you will find necrosis, that is centrilobar necrosis. So in sheep, if the dietary copper concentration, if it is more than 25 milligram per kg, it leads to chronic toxicosis. And if again, as I said, either copper or molybdenum, if the copper is less than 8 milligram per kg, that may be harmful. If molybdenum concentration is 0.5 milligram per kg, so copper molybdenum ratio is to be taken care of. So if you come across a copper toxicosis case, so what will be the approach for treatment? First, as we know, whenever there is a toxicosis or poisoning case, we have to increase the excretion of that poison which is inside the body. You give a huge amount of fluid so that the fluids are flushed off, so the tissues are flushed off. Then ammonium tetrathiomolybdate, that is available as injection, and it can be given at the rate 1.7 milligram per kg body weight. Intravenously it can be given, or it can also be given subcutaneously, double the dose, so that it is absorbed slowly through the circulation, and the excess copper is removed. See, this ammonium tetrathiomolybdate, it removes the copper from the intracellular space, from the lysosome and from the cytosol, and it gets bound to this, the copper gets bound to this ammonium tetrathiomolybdate. It replaces the molybdenum, and it the copper gets attached to the molybdenum part. So there is exchange, and that will cause exchange of the copper to the molybdenum, and it will be excreted, so that will be eliminated. And the withdrawal period for this should be approximately 10 days. So by that time, the sheep should not be slaughtered, or the milk from the goat should not be consumed. Again, there is another chelating agent, that is D-penicillinamide. That is to be given at the dose rate of 50 milligram per kg daily for 7 days. And again, or calcium versenate, it is also available in the market, that can also be given in powder form. This helps in excretion of the copper. Then daily oral administration of ammonium molybdate, you can give 100 milligram, or sodium thiosulfate. Again, as I said, copper, molybdenum, and sulfur. This copper, it exchanges itself with molybdenum or sulfur. So either you give molybdate or you give thiosulfate. So that will exchange the copper with these ions. And that sodium thiosulfate can be given at the dose rate of 1 gram, and animals that can be given for 3 weeks, and this helps in preventing the hemolysis and it avoids the toxicity. Dietary supplementation of zinc acetate. Zinc actually it acts as a protectant and it helps in tissue regeneration. So that can also be tried at the dose rate of 250 parts per million. That helps in reduction of the absorption of copper because it acts as a coating on the mucous membrane. Vitamin C can be used as antioxidant and that reduces the damage of the RBCs. As we have seen in corona duration or in any disease, vitamin C is a very good antioxidant and it should be preferred in most of the diseases. Wherever you find some hemolysis or you find any viral diseases, vitamin C can be used very well. And in case of sheep, it is 500 milligram per day and can be given as injection or it can be given orally also. To remove the toxic effect and to remove the excess amount of copper, we can flush out the body and tissues. It can be done with the help of fluid therapy. And along with that, fluid therapy, you should also maintain the diuresis, because again, if you're going on giving fluids and the diuresis is a problem, or the amount of fluid which is given is not filtered properly, or the amount of fluid is not in comparison with the amount of fluid excreted, urine output, so that will cause extra fluid in the vascular compartment, and that will lead to hypotonicity, and that will again, instead of causing any benefit, it will cause a damage to the animal. So renal, it will again cause renal damage, it will cause hemolysis, it will cause RBC turgidity, and it will cause rupture of the RBCs. So maintaining the copper content of the diet at the dose rate of 7 to 11 parts per million, and then also achieving the copper to molybdenum ratio of 61. It is helpful in preventing copper toxicosis. Otherwise, the copper toxicity is at a level when the copper and molybdenum ratio is about 101. Okay, so primary, again, there are some phytogenous roots also from where the animal is getting copper, and it may also lead to copper toxicosis. Top dressing, many times we are doing top dressing. So when there is excess amount of copper in a particular area, in a particular pasture, you can also go for top dressing of the pasture with molybdenum. That is 1 ounce or 70 grams per hectare. You can dissolve and you can go for a spray. So that will also help in reducing the absorption of copper. This is one of the plant toxicosis where you find hemoglobinuria. So in India and in many regions of the world where cotton is being produced, the cotton seeds, they are used as oil cakes for animals. They are used as source of protein when the crude oil is extracted, and that is used for some cosmetics and some medicinal industries. But when the oil is extracted and the remaining portion is there, it is rich in protein, so it can be used as concentrate, and it is used as concentrate in many areas. So gossypol, the cotton plant, it has glands, and the if the sheep and goat, they are let loose to graze on these plants, or if the cotton cake is being fed in huge amount to the animals, then there is toxicity. Usually this is toxic in case of animals in sheep, like ruminants, when the rumen is not properly developed. Okay, in case of young animals, so this is more common. So after consumption of the cotton seed, there is excess amount of free gossipol in the blood, and then that will cause hypertoxicity in the hyper amount of gossipol in the blood, and that will lead to toxicity. Now what happens, the affected animal, there will be hematologic changes, and again there will be increased RBC fragility that will lead to lysis of the RBC, that will lead to anemia, that will lead to icterus. And again, the gossipol also has some effect on the heart tissue. So there will be cardiac failure and sudden death. This will also get absorbed in the liver, and there will be liver necrosis, and there will be reproductive failure, because since there is anemia, there is cardiac failure, there is cardiac affection, there is liver affection. So all these things will lead to reduced circulation in the deeper part of the tissues in the body, and also that will cause, because there is lysis of the RBCs, again there will be anemia, and all these things will lead to reproductive failure in case of animals. And along with that, certain researchers have shown that it affects the spermatogenesis and libido in case of males, and in case of females there will be irregular cycling, there will be irregular cycle of ovulation, and if at all there is pregnancy, there will be embryonic death, and there may be abortion. So the only treatment here we suggest is removal of the cause, that is removal of the gossipol from the feed, and give some supportive therapy. So, how to diagnose this? So, again, a history that the animal was exposed to the cottonseed meal, or the animal is being fed for a long time on cottonseed meal or products, and then the clinical signs, that is, there will be sudden death and anemia and all, and then there will be reproductive failure. And again, if you see these steps, and many times in the field, we do not even go for laboratory diagnosis uh, because of unavailability of facility. We don't go for diagnosis, we simply see the symptoms and we go for treatment. So most of the time we are treating with antimicrobials and supportive therapy. So one differential diagnosis here can be that, like, if the animals are being treated with antimicrobials and still the animals are not recovering, the animals are not showing proper growth and all, so you can suspect that there is some phytotoxic effect, and there is some plant or plant product which is causing toxicity. Then post-mortem you can find that there is cardiomyopathy and hepatopathy, and there will be tissue fluid accumulation. And along with that, if you go for sending the tissue samples to the toxicology laboratory, you can see that there is increased concentration of free gossipol in the tissues. And also, if you go for analysis of the feed, you can also find high concentration of gossipol in the feed or in the diet of the animal. Then how to go for differential diagnosis? So complete blood count is to be seen. If there is reduced amount of RBCs and other things like WBCs and all, they are normal, then we can suspect that there is some non-infectious cause, because in infectious cause definitely the WBC picture is going to be high. Then you can also go for blood picture examination, blood slide preparation, and then staining these slide. So you can go for identification of some intraerythrocytic protozoan parasitic infection if it is present, or there are some toxic bodies present in the erythrocyte or remnants in the erythrocytes. So you can go for identification of that. Then you can also discriminate whether this is a regenerative anemia or non-regenerative anemia. If you find more number of reticulocytes on the smear, then you can definitely say that this is a regenerative anemia. Then a serum biochemistry panel can also be tried. You can go for liver function test, then kidney function test. Then you can go for estimation of the different elements in the liver and tissue, especially as I said that these are the important causes of anemia, hemolytic anemia. So you can definitely go for estimation of nitrates and estimation of your copper, molybdenum and all. This way you can find out the trigger of hemolytic anemia. Then plasma hemoglobin concentration, that is definitely increased, and because of that there will be because of intravascular hemolysis, the plasma hemoglobin concentration will be increased. Then serum LDH concentration, that will also be increased because of damage to the kidney, heart and liver. So there will be splenomegaly. If there is splenomegaly and there is absence of intravascular hemolysis, and there is presence of extravascular hemolysis, then the cause should be no. Extravascular hemolysis, there will not be splenomegaly, and if there is intravascular hemolysis, there will be splenomegaly. Then you can also go for dipstick test. Urine dipsticks are available, and you can just take the fresh urine, midstream urine you have to collect, and then you can dip the test strip, and you can check the color change, and you can correlate with the colors on the particular dipstick box. So they give a color index, and accordingly, correspondingly, you can go for checking of that, and then you can find out whether it is a hemoglobinuria, whether it is a myoglobinuria, hematuria, or some other reason which is causing urine to oxidize, and thus it will cause later on when this urine is kept standing for a while, for 15 minutes or half an hour, the urine will change color from yellow to dark yellow, and sometimes that is very dark, that it seems like it is a hemoglobinuria. So iodine, bleach and all, they are to be checked. Then how to differentiate between hemoglobinuria, myoglobinuria? For this you evaluate the plasma color, then you go for hemoglobin urea. Then there are certain tests wherein you can discriminate between hemoglobinuria and myoglobinuria. So these are the different colors. The extreme right side, it is a normal urine, and then the second one, it is little slightly darker. It may be because of some amount of hemoglobin, or it may be because of some oxidizing agents in the urine. The latter three, from the third to the extreme left, they are hemoglobin urea, the hemoglobin urea, or they may be myoglobin urea. Okay, so I will just tell in short how to discriminate between myoglobin and hemoglobin. This, the extreme right, this is a case of hematuria, and the urine while collection, it was like the reddish color which you can see on the left side test tube. Urine when you collected, it was like that, and when you go for centrifugation, you see that the urine color was actually yellowish color, but there were huge number of RBCs which have settled down, and this yellowish color of the urine on top of that button of RBC, that is because of the rupture of certain RBCs when it was standing in the urine, because of the pH of the urine. The some amount of, or some numbers of RBCs, they are rupturing, and that will impart some yellowish or orangish color to the urine. Okay, so this is a case of hematuria. So sample before centrifugation and sample after centrifugation. So this is how you can discriminate between hemoglobinuria and hematuria. So true hemoglobinuria, it causes deep red to brown discoloration and gives positive reaction to the biochemical test for hemoglobin. You can take a few drops of urine on the filter paper, and you can put ammonium sulfate, and you can see if there is change in color. You can say it is hemoglobin urea. So false hemoglobin urea can be seen in case of hematuria, as I've shown in the previous slide. Hematuria and hemoglobin urea. Hematuria, the RBCs, they settle down, And in case of hemoglobinuria, the RBCs, they are lysed and hemoglobin is released. So this can be detected also with the help of microscope. You can take 1 or 2 drops of fresh urine and you can put a cover slip and you can see under the microscope. RBCs can be seen as a reddish dot in the under the microscope. Now, if there is no evidence of hematuria or hemolysis, myoglobin can be considered. So, how to test for myoglobin? You take a urine sample, myoglobinuria, by urine precipitation with the help of 80% saturated solution of ammonium sulfate. So you take 2.8 gram of ammonium sulfate and it should be added to 5 ml of urine, with a neutral pH. So after you mix it, you centrifuge this mixture, and if the supernatant, if it remains dark, then it is a confirmed case of myoglobin. You can also do this test. You can take this saturated solution of ammonium sulfate, take a filter paper, put few drops of urine, and then you add this ammonium sulfate. So in case of hemoglobinuria, you will find small spot of dark red color at the center, and the periphery it will be yellowish color. But in case of myoglobin, the whole thing will be reddish in color. Sometimes there is interpretation problem, and myoglobinuria, it requires also we can see for some muscular damage, or there is some myositis, and along with that you can also go for seeing the serum creatine kinase activity. If it is high, then definitely muscles are getting damaged, and there will be increased amount of myoglobin, and that will be drained out in the urine.